I would like to talk about DNA modifications, and this field kind of got very, very complicated, so it's maybe good to remember the basic question, which is basically, why are some sites methylated, CpG sites, so cytosines methylated, dark spots over here, and some are not? And does it matter? So early on, the idea was proposed that there are some de novo methyltransferases that de novo methylate CpG sites, and then there are some enzyme that maintains it after DNA replication. That would be the DNMT1. So does it matter? There was a lot of data out there indicating that there were changes in DNA methylation correlating with development and cancer. But it was all correlation. So we early on set out together with Rodienis' lab to test this. We produced a hypomorphic allele that produces less DNMT1, causing hypomethylation. And there we indeed found that it affected the development. These mice are runted and then developed very aggressive tumors. So clearly establishing that DNA methylation is required for normal development, for genome instability, causes genome instability in cancer. By the way, it also keeps in check, as you might have guessed, the elements that we just heard about, the retroviral elements. So it clearly matters. And if you take a look at changes during development, you see over here dramatic increase in methylation during gametogenesis. And the first thing we did in our life was to erase all the epigenetic information that we inherited from mom and dad, to then reaching a low point over here at implantation stage. That's where the pluripotent embryonic stem cells are. From there on, we establish our own DNA methylation patterns and then maintain them over long periods of time, over many cell division cycles. So if you want to also reprogram cell fates, you need to overcome these epigenetic hurdles. So how is DNA methylation, how are DNA methylation patterns efficiently maintained over cell division cycles? And that is the DNMT1, that's the ubiquitously expressed methyltransferase that recognizes these hemimethylated sites and methylates the cytosines in the newly synthesized strands. And in fact, the replication of epigenetic and genetic information is physically coupled, as we found out. The DNMT1, it's a very big protein, has a catalytic domain over here and a huge regulatory domain. And it physically associates with replication machinery through association with PCNA, which is the central loading platform of replication via this PCNA binding domain. And back a long time in my postdoc time in Tim Baster's lab, I mapped this additional region over there that targets DNMT1 to late replicating heterochromatin. And as that was all we really knew about it, we just called it TS for targeting sequence. And today I will explain to you why it took us so long to figure out how it actually works. And as you will see, it's a rather complicated, surprising story. So first of all, we and many other labs looked at interacting factors to learn more about the regulation of DNA methylation. And there are a number of proteins interacting with DNMT1. The green ones are additionally post-translationally modifying it. And you see over here the PCNA that I already mentioned. And the one I want to focus on today is probably the most interesting. It's UHF1, also known as NP95. And if you take, for example, here a look at a densely methylated region, the silence promoter, and it's in these embryonic stem cells, it's highly methylated, so over here. And if you knock out DNMT1, the major methyltransferase, then you lose most of this methylation, as you would expect. But the surprising thing, also if you knock out your HF1, you have the same dramatic loss of DNA methylation, although it's not a methyltransferase at all. So how can that be? So clearly we need your HF1, but how? So the idea was that maybe your HF1 is the one recognizing the substrate and then bringing in DNMT1. There's many ways to test it. Just show you one way we looked at it. Here you can actually see the binding of UHF1 at chromatin. You see these spots here. These are chromocenters, highly methylated regions in the genome. So you can see the localization over here. And if you remove all methylation, triple knockouts, knock out all CE methyltransferases, you see it doesn't really change the association of UHF1 with these chromocenters. However, if you knock out H3 K9 trimethylases, as in the SUVA 3-9 double knockout, so you lose all the localization, all the binding of UHF1. So clearly indicating that H3 K9 trimethylation plays a key role. And then now it's time to take a closer look at UHF1, and I'm kind of summarizing a lot of work over here. So it's a multi-domain protein. It has a tandem tutor domain that recognizes H3K9 trimethyl, a PhD domain that recognizes arginine 2, as an A domain that recognizes this hemimethylated region. And by systematic mutagenesis, we could show that all of these domains are necessary but not sufficient. In addition, you needed this ring domain, and ring stands for a really interesting new gene, kind of funny, not very informative, but you may know that it's a ubiquitin ligase. And what it does is it ubiquitinates lysine 18. And that, in turn, is recognized by, remember, the TS domain. We found out that it contains a ubiquitin interacting motif, so it recruits DNMT1 and at the same time allosterically activates this catalytic domain to now finally methylate the DNA. So you see, UHF1 is kind of bridging histone modifications and DNA modification. It reads epigenetic information. It also writes new epigenetic information. So we were pretty proud having found this lysine 18 ubiquitination. At the same time, our collaborators in Japan found out lysine 23 ubiquitination. We're pretty sure about this. This is one of the few occasions where both could be true, could be right, and that's beautifully shown in their crystal structure that shows that the TS domain kind of wraps around the histone tail over here and inserts a loop here in between these two ubiquitin residues. So both are ubiquitinates. It's dual monoubiquitination. That is actually the signal recognized by the TS domain. Then we looked at other targets of UHF1. Turns out that UHF1 also ubiquitinates PAF15, and again in collaboration with our Japanese collaborators in Nakanishi group, we could show that if it's again dual monoubiquitination, if you mutate these lysine residues over here, then you lose DNA methylation, and this time preferentially early replicating DNA. So we have now two pathways to recruit and maintain DNA methylation. And this is kind of putting it together. So I showed you the H3 pathway, so the histone tail recognized by UHF1, ubiquitinating it, setting a signal that is recognized by DNMT1 that then methylates the DNA. Very similar, kind of, is the PAF15, a PCNA-associated factor, and in fact, this tail has some sequence similarity with H3 tails, also recognized by UHF1, ubiquitinated, DNMT1, and so on. So we have two pathways, one probably preferentially operating in early S, the other more in late S. So two pathways. So when we look at DNA methylation patterns, as in here, we just tend to think like, how is this established, like methyltransferase putting the mark somewhere like this. But you could think of a pattern also be created by selectively removing it at some sites. And there's a reason why this for a long time wasn't really considered DNA demethylation, because the methyl mark is a very, very stable mark. So it is this carbon-carbon bond, and from metabolics, you know that in order to break it, you have to oxidize. So the field was very excited when in 2009 2 papers came out showing that indeed a methyl group can be oxidized by TET proteins. We know now that the TET proteins can oxidize methylcytosine to hydroxy, formyl, and carboxycytosines. So we looked at, so it is basically modifying the modifications, creating new epigenetic marks. So we looked at proteins that would recognize these new epigenetic marks. It's an entire list, of course. One of the interesting candidates binding to formyl cytosine preferentially was TDG, thymidine DNA glycosylase, that excises this oxidized base and together with base excision repair replaces it by unmodified cytosine, so it's an active demethylation pathway. So here it is shown if you offer different substrates, you see that TDG preferentially excises formyl cytosine and carboxycytosine even much better than its original substrate. You know, it was described as thymidine DNA glycosylase, recognizing this GT mismatch. So if you look again at this and wonder what would happen if we knock out TET, well, you would expect to get, of course, more of this and less of that, right? And that's exactly what you're getting. So more methylcytosine, of course, and less of the oxidized. So far, so good. But most of these changes occur at sites where TET is not bound. So kind of suggesting in the CS cells that there are indirect mechanisms. To get an idea what these indirect mechanisms could be, we looked at effects on gene expression. So if you knock out TET1, you get, surprisingly, some genes that are upregulated and others that are downregulated. So TET has actually a very complex role. So it works in activation and in repression both. I should also mention that TET has non-catalytic function, but that is an additional story. So if you look at these gene expression changes, one gene that really stood out was DPPA3. So it clearly needs TET1 for expression. And if you look in the literature, DPPA3 is also known as PGC7, primordial germ cell 7, so it's expressed in primordial germ cells, also known as STELLA. And the literature is a bit confusing. There are lots of kind of conflicting results. So interesting, DPPA3 is expressed at phases of global demethylation. So it's directly, as I already mentioned, after fertilization, there's this global wave of demethylation. That's where DPPA3 is expressed, and also in primordial germ cells, where also some demethylation occurs. And interestingly, at that phases, your HF1 is in the cytoplasm. Get an idea what's going on, we looked for interacting factors. So we found that DPPA3 is interacting with the maintenance methylation machinery with DNMT1 and UHF1. So far, so good, makes sense, reassuring. Then we found all these proteins here in LILA, and these are factors of the nuclear import-export machinery, kind of suggesting that it is shuttling between inside, outside the nucleus. So that brought us to the following possibilities. So how could DPPA3 affect DNA methylation? So first of all, it could compete with UHF1 for binding, kind of compete it out of the chromatin. Then it could form a stable complex with UHF1, and it could also export UHF1 out of the nucleus, as we have seen in these pictures I just showed you. So I just wanted to show you one way we looked at it, two ways actually. So what we see over here again is UHF1. You see it bound to chromatin, these heterochromatin foci. over time. And if we now in these cells express DPPA3, what you see is that we kind of displace UHF1 out of the chromatin. You see this pattern dissolving and shoveled to the cytoplasm. And this mobilization, this displacement out of the chromatin, you can also see by FRAP. As you see over here, normally UHF1 has a slow recovery, kind of reflecting transient association with the chromatin. And if you express DPPA3, you have this dramatic mobilization. So can we directly test whether UHF1 forms a complex with DPPA3? There are, of course, many ways. I want to show you one way. It's kind of fancy. Rick's raster image correlation spectroscopy. So basically what you do is you scan your sample with a confocal as normal, and at each point in the confocal volume you record the fluorescence, the red and green fluorescence. So if you have UHF1, DPPA3 labeled red and green, you look for the correlation. So if they are in a complex, then there is a high correlation in the red and green fluorescence because they come in the focus. Signal goes up, move out of the focus, signal goes down. So there's a high correlation. So you can plot this over here. So over here. To get an idea for this kind of method, if you just look at GFP, green and red fluorescence, fused, so constitutively together, you see a strong correlation because fusion, they're always together. If you express them separately, so they move separately, this is kind of the background signal. And here, our fusions, UHF1, DPPA3, you see a clear correlation signal over here. If you introduce a mutation disrupting this interaction, you lose this correlation signal. So that is one way to look at it. Then do we need nuclear export? So it's time now to look at DPPA3, this teeny tiny protein. It's just 159 amino acids, no real structure to talk about. It has here this conspicuous nuclear export signal, kind of fitting this import, this shuttling inside-outside the nucleus. So do we really need that? So we introduced the mutation. And you see now it is not shuttled out of the out in the cytoplasm, but still it effectively displaces UHF1 from the chromatin and affects the demethylation. And so clearly arguing that we don't need nuclear export. So clearly it is this kind of complex forming that explains the function of DPPA3. So we took a closer look at it, and together with the Arita group in Japan, together with the Nakanishi group again, we could show that DPPA3, which is an unstructured protein, it binds here to the PhD domain, and in binding forms this helical structure over here. And there might be additional interactions, contacts that we didn't see in this structure. So clearly, DPPA3 forms a complex with UHF1. So pretty amazing, this teeny tiny protein that has the power to erase methylation. So it's time to take a look at the evolution of the epigenetic system. So you see over here, the first enzyme that was showed really up was DNMT1, then followed by UHF1 and TET. And DPPA3 we found only in mammals. So it is a really recent recent development in evolution. So we find it here in mammals. And that correlates with the occurrence of this post-fertilization demethylation wave, because we find that only in mammals and not, or at least not as pronounced in the other vertebrates. So in other words, frogs don't have DPPA3 and don't have this post-fertilization demethylation wave. So we contacted our friends in Tokyo, Nakanishi group, and the idea was, with one protein we could turn a frog into a mouse. So we sent the DPPA3 to Japan, and they tried it in the frog system. So Atsuya tested it in his system, and he could see that also in frog, DPPA3 displaced the maintenance machinery out of chromatin, shown over here, not the replication machinery, important control. Then he did the methylation assay, and he could see a complete inhibition of methylation just with this single teeny tiny mouse protein. It was so dramatic that he couldn't believe his results, and he repeated it with fresh solutions and could confirm it, of course. So also in mouse, in frog, DPPA3 can completely block DNA methylation. So kind of we could transplant the demethylation phenotype with just one protein to frogs. So kind of emboldened by these results, we got daring and went back another 100 million or so years in evolution and turned to fish over there. And there we collaborated with the Takeda group and in Japan. And Rio High looked at Medaka fish over here. And you see also fish is highly methylated. All these spots here are nuclei containing methylated DNA. And if they express the mouse DPPA3, you see they completely erase DNA methylation. It's really impressive. So to summarize it all up, what I showed you is that TET oxidizes methyl groups, and these oxidized forms are recognized, so that creates a new epigenetic signal. And it is also recognized by TDG that excises it, followed up by base excision repair, replaced it with cytosine, so it's an active demethylation. That kind of makes sense if you want to demethylate specific sequences or activate specific genes. But of course, it comes with a risk because it involves a repair process. However, if you want to just globally demethylate and reprogram, then this passive demethylation might be the better choice. And I showed you that TET1 drives the expression of DPPA3 that then inhibits the maintenance methylation machinery. I should also mention that TET also has non-catalytic function. So if you just it's a difference whether you just mutate the catalytic activity or whether you knock it out altogether. It turns out that TET interacts, for example, with SYN3A complex and also plays a role in suppressing retroviral elements, but that is another story. Another important question is, where does DPPA3 actually come from? And that also brings us what we heard already about this arms race between viruses and normal development. Turns out that yes, global demethylation is good for reprogramming, of course, in the beginning of our lives. But of course, it comes risk because it activates also some elements. And turns out that we kind of learned to work with, to live with them. And in fact, early development employs some of these retroviral elements for early regulation. But that is yet another story. So now, would like to acknowledge the three main characters, Sebastian Bultmann, Christopher Mulholland, and Wei Xin, that were kind of the driving forces in the results that I showed you, and the collaborators that I mentioned throughout my talk, and I thank you for your attention.